So I'm Lea, I'm a third-year PhD student, and this is already my third time speaking at this workshop, so I really appreciate this. And I'm going to talk about what I've been doing in the past years today, about my PhD project, which is, no, this is unfortunate. Let's try like this. So you see the presentation, correct? Because then...
Yes.
Perfect, good. So the project is about evolutionary trajectories. So we want to see how the minimal cell evolves throughout evolution and really track the changes that it makes during this process. So we have two questions that we want to answer. The first one is, how repeatable is phenotypic evolution? And the second one
Leah,
is,
sorry, I still only see your first slide. Are you on the first slide or the second slide? Okay, now I see the second slide. Great, great.
This is unfortunate because now I cannot, you see the complete presentation and not what I add in the meantime, but we can make do like this. But yeah, this is unfortunate. I'm not sure why it's not working. Good. So you see now the complete slide, and I will just push this away for a second. And so basically it's how repeatable evolution is and how predictable evolution is. And then we want to compare the predictions, which will be growth rate, reaction fluxes, and metabolite and protein concentrations. So in the end, we want to compare these two together. And the second part is done by my colleagues from Düsseldorf, which are Charles, Hugo, and Peter. So I'm going to be focusing on how repeatable phenotypic evolution is, so on the wet lab part, which I'm going to present today. So this is what we are planning to do. Basically, we use the minimal cell, and we want to use it as a model organism. And the main advantage for us is that it has minimal regulatory elements, as we heard today also, which means that we hope that during evolution, even if like a regulatory gene is mutated, that it doesn't affect a lot of parts of the other genes. And this is why, this is what happens in E. coli, for example. So we really want to see the direct changes due to our selection pressure. Also because of this, we hopefully have reduced phenotypic plasticity. And we saw, though, that the minimal cells are able to evolve. And because of their small genome, they are good for our modeling approach because it's rather complex. So we want to start very simple. So what I then had to do is establish the minimal cell in our robotics platform, which we use for our evolution experiments. And this robotics platform has one major advantage, which is that we can use a large number of replicate populations because we have a stack of 96 well plates. We can also keep the cells in exponential growth. So we have a nice fitness correlation because the cells never enter stationary phase. We do multi-omics, like I said, and we ideally want to use a defined medium, which a student assistant of mine is now working on, Neka. And it's been really nice to see how many people are currently working on the defined medium. So this is our evolution robot. Just to show, it's a very big machine. It's in a room at 30 degrees in our basement. It has an incubator and a lot of space for plate storage, so we can really do a lot of plates and plate reader, everything that you need to measure growth. And then this is the heart, the links, the liquid handling system, which basically does the dilution for us. So every couple of hours we can dilute. And then we had to optimize the system for our cells, for the minimal cell. And because the system originally was developed for E. coli, so we needed to adjust more specifically the medium. And I'm just going to quickly show a bit what we had to do because, as you probably know, the medium is quite bubbly. So we had a lot of false OD600 measurements because of the air bubbles, but we just switched to reverse pipetting. So we now have no issues anymore. And we also faced a lot of clogging because we have to wash the tips multiple times because we use them for a day. And during a day, we have about 6 dilutions. And so the pipette tips can be washed very easily. and we just washed them directly now after use, and everything is fine. So this is what we are now working with. I'm gonna push this aside. So this is an example evolution experiment that we performed so far, spanning 100 hours and about 12 replicates, you can see here. And it uses the protocol that I've explained before, that we incubate the cells, we read the optical density every 25 minutes, so we have real-time growth tracking. We then calculate a target OD, a target start OD, which is here 0.01, and then we perform dilutions. And these are done, like I said, so we can really nicely see that the cells are growing in an exponential phase, a very nice and straight line. Then we dilute, they start growing again, so they never reach stationary phase. So now that we have established the robotics platform, we want to find suitable selection pressures. And because what we want to do is more like a fundamental approach, we didn't really care about what exactly the selection pressures are, but more like what can easily be done in our robotics platform and what is nice for the modeling approach. So the first thing that we wanted to use is 2 different starting strains, and we got by John kindly from the JCVI, we got 2 starting strains, the SYN3B and SYN3 plus PDHA, PDHB, which codes for the E1 complex of the pyruvate dehydrogenase. So in theory, the cells now have a complete PDH, and in theory, they should now be able to ferment also acetate from pyruvate and not only lactate, as you can see here. So maybe we see some alternate metabolism happening during evolution. The second selection pressure that we decided on was basically the first one, doing nothing. And here you can see just a synthesis of the 3 evolution experiments that have been performed so far with the minimal cell. On the left, Mugareisha et al., who measured relative fitness. On the right, Hossain and Sandberg et al., who measured growth rates. And they performed experiments over different amounts of generations and really could nicely see that the fitness is increasing during evolution. So we can say the cells probably are not in an optimal state already, because genome reduction probably led to not everything being optimal, and also every evolution in the lab leads to an increase in growth rate if the cells are not adapted to the lab already. So we know we can just do nothing, let them grow in optimal conditions, and we will probably already have some nice results. The difference here to us is that we will leave them in exponential phase and not grow them to stationary phase. The second selection pressure that we decided on is temperature, and because the room the robot is standing in is at 30 degrees, we can just keep one cell in the incubator and one plate in the room, and the growth rate is about half. And Charles did some calculations on how this would affect the evolution based on enzyme kinetics based on KCAT values, because not only do the KCAT values halve, but they reduce in different ratios based on this estimation in this equation. So we have different ratios of enzyme kinetic reduction, which could then, in theory, lead to alternate metabolic adaptation and not only reduce adaptation in general. The third thing I want to look at is pH, and the reason for that is that we also got cells from the Saints lab when I visited them, and so we got cells from the JCVI which are fresh after genome reduction, and we got cells from Dresden which have been cultured in the lab for some time already. We don't know for how long, but basically some time. And what we did here was to throw the cells in a bioreactor and measure turbidity and pH over time. And what we can see is that, first of all, lab evolution caused an increase in growth rate, also an increase in yields, and a much higher drop in pH, which we see in the exponential phase, but also in the stationary phase. So we can assume that the cells produce more lactate, and this drop here seemed very like a lot to us. So we wanted to know if the cells are in a way hindered by this pH drop. And we sent the samples in stationary phase to HPLC analysis, what we can see here on the top left. And basically what we see is that after stationary phase, not all of the glucose is consumed. And on the other hand, if we put them in a bioreactor, but we add an additional NaOH feed, so the pH stays constant, all of the glucose can be consumed and a higher yield in theory could be achieved. So we know that the cells are definitely stressed by the pH in stationary phase. And then I did also some pH studies on how much pH can be tolerated, which you can see here. I just measured optical density and then calculated growth rates and yields. And I also put, from the slide before, you can see the medium end pH of the cells from the JCVI compared to the ones from Dresden, so the evolved ones. And basically, the cells we got from the JCVI, they have an end pH which is definitely above their tolerance, but the ones from Dresden, they end up at an NPH of 5.5, which they do not grow in anymore. So probably there's some sort of limit to adaptation and glucose consumption that is going on here. And then we also decided to just also let them grow in a reduced pH of about 7.8, which leads to 50% reduced yield and growth rate. So in summary, we now have an established platform for high throughput and evolution experiments with the minimal cell under defined and constant environmental conditions. And we will evolve them in different selection pressures soon. I wanted to start already, and I was hoping that today I could present the first initial results, but unfortunately we ran into a problem, which is that we want to start from a colony, basically a strain which is as homogeneous as possible. So we streaked our cultures on agar plates, and then I just picked colonies and let them grow. But unfortunately, these colonies grow much worse than our original culture. So I'm not sure what is happening here, if they have some kind of stress due to streaking, or I'm in stationary phase. And I was just, I just wanted to put this in the end if someone has some ideas or recommendations. And with this, I want to thank you and the lab I've been working in and all the help I got. And I want to apologize that you couldn't experience the presentation as I hoped, and I hope it was still fine.
Very nice, Lea. I like to see how your work is progressing.
Thank you.
And it seems like you are on the verge of being able to do some really terrific things.
Yes, I hope so. I'm very, very excited to start, and it was super sad that we ran into these issues with the colonies, and I would really appreciate some insight if anyone has some similar.
I am mystified about your colony issues. So SP4 with knockout or SP4 with serum?
I am working with knockout.
Okay. I would send your results to Kim, or in fact, describe your whole conditions to Kim, and I will publicize this to the bigger community. It might be worth taking one of those colonies and getting a genome sequence on it and seeing if there's something strange, given that these are colony isolates. But I'm just surprised. How many have you looked at?
Right now, I'm running 30, but running right now. But before, I just picked 3 because I was very optimistic that I'm just picking one and then I will start. But yeah, it would be interesting to see if this really is an effect, and it might be interesting to check it out more, because.
And these are cells that have already been evolved, or are these straight out of our tubes that we sent you?
These are from you, straight out of the tubes, basically.
Yeah, that should that really surprises me. But let's set up a time to talk, you, me, and Kim. So make us an offer. Not for this afternoon.
No, for me it's already evening, so
okay.
Thank you. This would be great.
Okay. Anyone else today? No, An, you're up.
Yeah, just a small comment that we also have some problem with getting colonies. So what you should talk to our colleague Tess. She is actively working on this right now, and basically she find that if you don't streak them and you just dilute and spread them, it helps a little bit more. The biggest problem is that you have to make sure that you dilute them enough so you can actually get single colony, because they're quite small, and if they're together it will be hard. But yeah, we have someone working on that, so maybe I can forward you her email.
Yes, this would be. I think I have her on Slack, so but thank you. I will.
Oh, perfect. Then that will be easy.
Okay, James.
Hi, Lea, beautiful work. Just a related thought. When we start our experiments on the plate reader, we tend to first do a serial dilution in a 96 well plate. I guess that would be at the expense of more generations, but at least that way you can control for variability in the initial number of cells in the well by finding at which tube does the dilution. The lower dilution start to affect, say, the pH of the growth medium or some other metric. And related to this, I've also made some pictures of cells and colonies. And of course, they're present at an extremely high density. So, and furthermore, when I try to take a colony off an agar surface, there's still a kind of a residue in the agar. I guess the mycoplasma somehow penetrate into the agar to some extent. So all to say, I've never tried to measure if you take a sterile loop and wipe the surface of an agar plate and then immediately measure number of colony forming units, both because of the aggregation as well as because of the cells that have penetrated into the agar. How many colony forming units would you expect? But I could imagine there being huge variations over perhaps even orders of magnitude from one well to another. But in general, beautiful work, and I'd love to learn more. Maybe I can reach out to you when you have time to learn more about the growth, because it seems that the growth rates you've achieved on the plate reader are more in line with what John and others have measured compared to our measurements, in which so far the doubling time seems a bit longer. So anyways, great work. Thanks for your talk.
Thanks. Of course, I would love that. And I've noticed too, for us, the growth rate on the plate reader seemed to work really well, measuring growth rates. And also, I didn't mention this, but regarding the colonies, it was really interesting because then I wanted to see if they regain their growth rate after I cultured them for a while. So I already tried some initial evolution, but they really don't seem to recover that fast, the growth rate. So it's really a mystery to me if there's some kind of mutation. If maybe perhaps I just picked 3 colonies that are mutated, but yeah.
Okay, thanks. Interesting.
Rosanna, you have a question?
More of an appeal with all of this work. I feel like I'm kind of the odd duck in a lot of ways with a lot of the physicality of the things that we are working on, although I see more and more of that. And I just wanted to make a broad appeal for physical measurements of things like mass and sizes of nucleoids and where things are. So if you don't mind me making that plug, John, that's one of the biggest things that we need. We're going to get more well-defined than that. I would like to actually think about a tag-along session sometime where we just have open conversation about that, if that's fine.
Excellent. We will hopefully a few minutes of discussion at the end that we can bring this up again.